Greetings, scholars, and welcome back to the academic track of the Fall Enrichment Program. If you like to eat, as I see that many of you do, then you will want to become more knowledgeable of the potentially serious global threat that motivates our invitation to today's speaker. And we're very grateful to get Dr. Jean-Marc Bonmatin on relatively short notice for this topic, and he's in between many international meetings that have their importance from this topic. Dr. Jean-Marc Bonmatin has a presence at researchgate.net that you may wish to consult, as I did this morning to anticipate his presentation. A June 2014 editorial in the journal Environmental Science Pollution Research proposes tightening the routine agricultural use of persistent and potent neurotoxic insecticides because of their interactions with the ecosystem long after their effect on that current season's crop is past. This is an open question with serious consequences about the effect of these persistent neurotoxins on pollinators and other non-target species, and whether they may be responsible for the colony collapse disorder in bees. And much more than honey production is at stake. Dr. Bonmatin is a researcher for the Centre National de Recherche Scientifique, the CNRS in France. He completed his thesis in 1987 and started out as a physicist, which then led him into chemistry, which then led him into biology, and now into economics and toxicology and all these things with agricultural impact. So you can never tell students where a very particular thesis topic may take you. His thesis topic was on the penetration of peptides, in particular bee toxins, bee venom, through membranes. And now look where you end up. So that's, I think, a life lesson that goes along with the particular content of today's talk. After graduating in chemistry and physics, he worked at the National Research Council of Canada on the dynamics of now cholesterol in membranes. He joined the CNRS's Centre de Biophysics Moléculaire in late 1989, where he started his research on the structure-activity relationships of various natural toxicants, antibacterial, antifungal, neurotoxins, etc. And from 2008, he's been involved in the European programs on colony collapse disorder. He's also involved in risk assessments for pollinators in several public agencies for France and for the 34 countries of the OECD. So thank you very much for investigating our bee potential on the KAUST campus and for letting us know more about this coming crisis.
Thank you, Mr. Chairman, for this nice introduction. I'm very pleased to be here, invited by KAUST for giving to you this lecture. You will see that this lecture mixed chemistry, physics, biology, toxicology, and history as well. So today I am going to speak about honey and bees, and especially about the decline of pollinators, which reveals a major environmental threat worldwide. So this is the summary of my presentation. We will have a little bit of epistemology, history about honey and medicine. We will speak about the colony collapse disorder, then about exposure of bees to systemic insecticides, effects of these insecticides on the model insect Drosophila melanogaster, then the impact of bees, of course. Then we will see our work we made, which is called World Integrated Assessment, and the effect of these systemic insecticides on non-target invertebrates as well as on vertebrate wildlife. So to begin, I would like to show you a few pictures rapidly. The first bees that are seen in the history of humanity came from ancient Egypt. Here you have bees, here, and this is probably the first beekeeper. So this is an old story, and bees produce honey, of course, for sugar, but also there is some conservation properties, and for instance, Hypothesis 4, here as an example, conservation of bodies. Virgil, the famous poet, is represented here in front of beehives, and in medieval era in Europe, this is the first picture showing beekeepers at work. I would like to show you also some famous scientists in the ancient world. The three first ones are well known, they are Greek, but the three last, Rhazes, Avicenna, Averroes, are Arabic. And they had a look on bees and honey. And especially this one, Rhazes, was the first known scientist, really a scientist, working on honey and bees. You have an example, he was a chemist, he was working with medicine and philosophy. And you have here something very interesting, the notebook, the lab book of Rhazes. He observed bees, he observed the property of honey, and he noted that, discussing these properties. This was the first one, and he wrote a book here about the benefits of honey. And in this book, there is a lot of things interesting, especially for chemists and physicists and pharmacologists. For instance, you have on these pictures two people working with honey and making some composed drugs. Okay? They are called teriacs. This is potions and receipts against people that are ill. Okay? For people that are ill. And they can contain more up to 50 ingredients. So for a chemist, this is really interesting because each time you have honey in the receipt. There was a lot of reagents and catalysts. Kinetics could be short, 1 week, or very long, 40 years. You have to wait 40 years for the receipt to be ready. And for chemists, this is just kinetics and catalyst. And you have the active compound and potassium. So we made several publications about this. Now, honey, nowadays. What is interesting is to see that there is as many kinds of honey as flower. You have two examples here. Honeybee foraging on sunflower and another one foraging on an apple tree. But there is also as many kinds of honey as bees. Because you can have a lot of bees, different bees. The main bees in Europe are Apis mellifera and Apis cerana. And you can have some hybrids, like this one, but fast. There is also as many bees as landscapes, of course, because bees are foraging on flowers in different landscapes. So now let's have a look on the properties of honey. The well-known properties are for asthenia and anemia because of the sugar content, glucose, fructose, maltose. Also some properties about the digestive system. Should be here. Maybe it's better. Properties about the digestive system because of the presence of enzymes, namely invertase and amylase. Also properties for otorhinorrhnology, it's difficult to say in English for French people, sleep disorders and cosmetic. These are well-known properties. These properties also are very important for blood circulation, antioxidant properties, diuretic properties, healing and birth properties, because it increases, honey increases the ratio of glutathione and collagen, so following the biosynthesis. More recently, we found some properties. The properties were well known, but not the molecules. and especially antibacterial properties due to inhibins and defenses. They are small peptides, okay, acting against bacteria, and especially Escherichia coli, Staphylococcus species, Pseudomonas species, Enterococcus species, and Bacillus subtilis. So honey is very important for medicine, in fact. Well, now I would like to present a problem linked to honey and honeybees. Here you have a graph of the honey production in France, the last 20 years. As an average value, we take the production between 1988 and 1994. And the average production was about 40,000 tons a year. This will be our 100%. And you can see on this graph the decrease of the production until 2009. And I have got the data for the last 4 years, and you can see that actually we produce 30 to 35% of the honey we produced before, 20 years ago. Why this production of honey is decreasing in France, but you will see it's everywhere in the world. This is a question of disappearance of bees. And this disappearance of bees is called the colony collapse disorder. And we will have a look on what is the colony collapse disorder exactly. I have the data from 2008 here, but it is exactly the same thing nowadays. You have the map of Europe and countries. And on each country, you have a circle. And this circle is simply proportional to the number of beehives. So you can see that the number of beehives in France, Italy, Hungary, Poland, and Germany is about similar. Some countries, Spain and Turkey, have quite more beehives. In each circle, there is a black sector. And this black sector corresponds to the number of beehives that died in 2008. So it corresponds in France, in all these countries, to about 1/4 to 1/3 of beehives which died in 1 year. And this phenomenon is occurring every year. This phenomenon is not limited to Europe. It occurs in North America as well as in South America. In fact, this phenomenon occurs in every industrial country where agriculture is intensive. There is some consequences, of course, not only on honey, but on pollination. For instance, animal pollination corresponds to 84% of the 264 crop species cultivated in Europe. And 74% of this pollination is coming from pollinators. And these pollinators are bees in these 74% cases. This represents an economic service of about 50 billion euros a year, every year in Europe. So, just remind that 71% of our food depend only on bees. If there is no bee, there is no food, no vegetable, no meal as a consequence in the cascade, and only cereals remain. Well, so the colony collapse disorder may have several causes that can act alone or together. And the main causes here are in red. Pesticides, especially insecticides, which are used in crop cultivation. Parasitism, you have an example here of a mite called Varroa jacobsoni, but also diseases viruses and other species inside the digestive tract. And also, in countries where agriculture is intensive, of course, the landscape changes and the intensive crop production produces a loss of diversity in terms of nectar and pollen, because bees are only feeding on nectar, pollen, and use water also. Nectar is the source of sugar, so energy, and pollen is the source of proteins. Well, it's a complicated chart here, but I just want to show to you some new pesticides. This one is pretty old, but all these ones are new. And on this slide, you can see the doses that are used per hectare. If we compare DDT with the new pesticides here in pink, you will see that the doses per hectare were divided by a factor of about 10. When you compare the toxicity of the new pesticides, and especially systemic pesticides, with DDT, you will see that the lethal dose 50, the dose that kills 1 bee over 2, 50% of bees, is quite decreasing, and the ratio is obviously 1 here, but can reach 5,000 here until 10,000. To conclude on this slide, I would say that we divided the doses by a factor of about 10, but the toxicity of insecticide increased by a factor of 5,000 or 10,000. So, how these new insecticides are used? They can be used classically as translaminar transfer on leaves by spray, or they can be used as seed dressing around the seed. The seed is sowed, the product, the insecticide, diffuses in the soil, then is taken up by the plant, and the plant is contaminated by the insecticide, okay? And a sucking insect, namely pest, would die very rapidly because of the poison. So now I would like to show you a little bit of chemistry. And here you are the main new systemic insecticides that are called neonicotinoids. They look pretty similar. Here you have the chemical name, and here is the name of the formulation, the commercial name. Some belong to companies, some belong to other companies, namely Bayer and BASF or Syngenta. So how do they work? I take the example of imidacloprid, which is known as a commercial name Gaucho for crops, for instance, sunflower, Confidor for fruit trees, Premise against termites, and Advantage for treatment of pets. So, imidacloprid is here. It's a kind of mix between nicotine and acetylcholine. And imidacloprid, as others, acts at the postsynaptic level in the central nervous system. It binds on the nicotinic acetylcholine receptor instead of acetylcholine, creating some hyperexcitation, and the insect will die in few seconds or few minutes. The particularity of these insecticides is that they are very, very active. And I would like to show you one experiment here dealing with chronic exposure of bees to imidacloprid. And at a very, very low level of exposure during 8 days, the mortality here increases and reaches 50–60%. So they are very, very active. So, what about the exposure of bees to these systemic insecticides? I used a unit which is really particular. It is 1 nanogram of toxicant per gram of food. For bees, food is nectar or pollen or both. So it's 1 billionth of gram of toxicant in 1 gram of food. Very, very teeny amounts. In CNRS, in my lab, we developed some experiment to detect such teeny amounts, such very low concentrations. It's a ratio of mass, in fact. And we were able to detect 0.2 nanogram of insecticide in 1 gram of pollen or honey. To give you an idea of this ratio, it's exactly the same ratio when we compare 1 cent of euro with the mass of the Eiffel Tower. That's exactly the same ratio. So, to detect such low amount of toxicant in pollen and nectar is really a challenge. Anyway, 0.2 nanogram per gram of toxicant of chemitoxam, for instance, correspond to 5,000 billions of molecules, which would be in a tumbler of water. So for the central nervous system of a bee, a small insect, this is really a huge amount. So a little bit of chemistry and analytics. We develop a technique for nectar and honey. And this is a pretty complicated technique, but however, starting from 5 grams of honey with this method, captures, we were able to purify the material, then to divide in 2 parts. I don't want to go into detail on this chart, but injecting the result in GC-MS-MS, 2 microliters, we were able to detect the systemic pesticides and some others. And we use also LC, liquid chromatography mass spectroscopy, to detect those pesticides. The results are pretty good because here you have the list of pesticides that we detected, and here you have the limit of detection expressed in nanogram per gram in the honey, and for deltametrin, which is a pyrethroid, we are able to detect 0.2 nanogram per gram. For fipronil, which is a pyrazole, we are able to detect 0.2 nanogram per gram, and for the nicotinoids here, we are able to detect less than that, 0.07 nanogram per gram. So we have now very sensitive methods to examine the exposure of bees. I can give you an example of results. When looking in field to real beehives and sampling twice a month the nectar collected by bees, we were able to see, this is the time here, this is spring and summer in 2009. We were able to detect a contamination by deltametrin here and a co-exposition with contamination with tofluvalinate here. And this contamination is not the same when you compare beehives. This one is not exposed, this one a little bit, and this one was really exposed to tofluvalinate. And this was a control area, the reference area, where there was no pesticide. And of course, while sampling all these nectars for analysis, we observed some typical sign of intoxication. Here you have a bee. It is not really foraging. There is some convulsive behavior, typically intoxicated. Now the results of this study. So here you have all the toxicants, and in pink you have the nicotinoids. Well, we found it just 5 molecules. Betametrin, clofluvalinate, acetamiprid, chacloprid, chemetoxam, and that's it. No, yes. Sorry. And here you have the maximum amount we found it in this nectar, okay, expressed in nanogram per gram. And we can compare these values to the maximum residue limit for human consumption, because nectar is producing honey. And you can see that in these two cases, we are far above the maximum residue limit for honey. In these two cases. But for bees, only few nanogram per gram are sufficient to kill the beehive. And what was interesting is that it's not exposure by one product, but by 4 products altogether at the same moment in spring, or 3 products together during summer. I spoke about nectar and honey. I can spoke also because we developed some analytics for pollen. In this case, corn was treated with chemetoxam, and we observed the presence of tertianidin, chemetoxam, and a little bit of chacloprid in this sample. And chemetoxam was present at 97%. Tertianidin is the first metabolite of this one, of chemetoxam, and we observe it at 35%. And the mean content in pollen was at 1 nanogram per gram. So bees are exposed by the nectar and by pollen. We developed also analytics for bees themselves. It's pretty bit difficult because metabolism in bees is very fast. But when comparing bees foraging on organic farming and comparing dead bees foraging on chemetoxam treated crops, we were able to detect chemetoxam here and its metabolite. And you can see that there is more metabolite than the parent compound. 0.4 nanogram per gram here in bees, 3.4 here for the metabolite. Well, you understood that there is a huge effect of the systemic insecticides on bees. But why? It's a challenge to understand because this huge effect was not expected. So we decided to work on the insect model Drosophila melanogaster because bees are wild animals. It's impossible to work with bees. You can't control all parameters. But with Drosophila, it's a good model to study toxicity. Rapidly, we exploit some Drosophila on larvae during a short time, 18 hours here, or a long time to imidacloprid, 8 days. And we compare the acute toxicity to the chronic toxicity. For the acute toxicity, we saw that females were more resistant than male. There is a factor of 2 here in the lethal concentration 50. And for larvae, there is a factor of 20 between the 2 values of these lethal concentrations. By chronic exposure during 8 days, we saw that the LC50 was sharply decreased. You have the 2 curves here. So this is the acute toxicity, mortality surviving Drosophila at the current situation here with a log scale. So this is the curve for acute toxicity, this is the curve for chronic toxicity. For female, there is a factor of 172 in the two toxicities. So how to explain that? In fact, I don't want to go into details, but the toxicity is a function of the dose, the concentration, but is also a function of the time, and time to the n power. And for bees, for instance, it's t squared. For Drosophila, we found that n, the power, the exponent, is between 1.6 and 2.5, which is the same thing as for bees. Note here that these bees have a t to the 5 power because they are pretty sick because of the presence of the Nosema pathogen. Another observation which could be important, this is the chronic mortality curves. So they are alive. All flies are dead here. But when we decrease the concentration, we found a new phenomenon. A new mortality appeared for male or for female at very, very low amount of toxicant. Here 10 nanogram per gram, here 1 nanogram per gram. This scale is expressed in nanomolar. And there was some differences between all these curves. We made some statistics showing that, of course, mortality depends on concentration, on the sex of flies, but also on the sex concentration interaction. Well, this was lethal effect. What about sublethal effect? We studied the fecundity of this fly by exposing the fly, the female fly, during 5 days here. So after mating, we counted the number of adults. And what is interesting is that the fecundity decreased at very low level of amount of toxicant, especially here. The surviving flies had a lower mortality by minus 16%. We observe also some strange behavior about mating. So we exposed both male and female during 5 days, and we counted the number of mating. And even at a concentration where there is no effect on the survival, we found plus 30% of mating at 0.1 nanogram per gram. And for insects, it's a reflex for surviving. All this was published, of course, and what was interesting is that if you compare the lethal dose 50 after acute exposure here and the lowest observed effect concentration, there is a factor here which is huge, more than 3 millions for males, more than 8 millions for females. For females. In other words, this means that there is no safe dose. You can divide the dose by several millions, you still have an effect. So now we understand better the biology and the toxicology of these products, and we are going to see some literature about the effects on bees. So here is the first paper showing that exposures of sublethal doses of fipronil and catocrit, two systemic pesticides, highly increased the mortality of honeybees previously infected by Nosema ceranae, which is a pathogen. So there is a link between intoxication and the presence of pathogens. All these two elements work together. They work together to kill bees, especially in the field. Another publication is very interesting. It deals with chemitoxam, a nicotinoid, and at very low level, the level you find in fields for bees, non-lethal exposure of honeybees to chemitoxam causes high mortality due to homing failure at level that could put a colony at risk for collapse. In other words, bees are not returning to beehive. Another publication was about bumblebees, because in France there is a lot of bees, in England there is a lot of bumblebees. Okay, so some English people were interested in bumblebees, and with imidacloprid here, they found it that treated colonies had a significantly reduced growth rate and suffered of 85% reduction in production of new queens compared to control colonies, of course. Another publication, very recent, from Di Prisco, my colleague from Italy, and this group shown that nicotinoid insecticides can impact the immune system. And especially, this system is found in all insects. So, nicotinoid can impact the immune system of bees, bumblebees, or all non-target insects. Another one. Yeah, sublethal exposure to nicotinoids impaired honeybees in mentioned before, proceeding to colony collapse disorder. The link is done. When you apply nicotinoids, there is the colony collapse disorder. This is an important publication, of course, and it was shown for imidacloprid and clotrimazole, two nicotinoids. And this last publication shows that, in fact, this is not only honeybees, this is not only bumblebees, but all pollinators that are affected and the services they provide. So we were worrying a lot about the impact. That's why the European Commission decided to ban some nicotinoids, these 3-1, imidacloprid, chemitoxam, and clotrimazole, for 2 years, but the Commission didn't ban this 3-1. This 3. Okay. Why did they ban 3 products and not all products? It's probably because there was also some other publication saying there is no problem. In this publication, you can see that authors conclude that this data demonstrates there is a low risk to honeybees from systemic residues in nectar and pollen following the use of chemitoxam as a seed treatment on oilseed rape and maize. So you have some publications showing there is already a problem and some others showing there is no problem. But if you look attentively to this publication, they are provided by companies. All authors work for companies. So it's a question maybe of conflict of interest. In this workshop, second example, in this workshop, authors founded people participating to the workshop found that nicotinoid pesticides were judged to be unlikely as the sole cause of this reduced survival of food and could possibly be a contributing factor to the death of bees. But again, if you look attentively, you will see that the workshop was paid by Bayer Crop Science, and every researcher in the workshop was working for Bayer Crop Science, or Syngenta, or BASF. I have another example here. And I just go to the conclusion. Sublethal effects of pesticides on behavioral learning and immunity are subtle and may not be measurable at realistic exposure concentration. And of course, this conclusion could be questionable. So, to make things very clear, we decided to make a world integrated assessment on these systemic pesticides, namely nicotinoids and fipronil, to see the real effects they have on non-target invertebrates as well as on vertebrate wildlife. And I will present you some publications about that. The starting point is that more and more researchers are aware on the impact of nicotinoids, and some are thinking that they are at the root of a global wildlife decline. So, we decided to investigate. Here is our chairman, Dr. Martin Willebed von Lixmon. He is an entomologist. He is not a chemist. He is not a physicist. He is not a biologist. He is just an entomologist. And at the very beginning, a group of entomologists observed that there was a global decline of insects. And the question was, why? So, I am going to present the World Integrated Assessment, and this is the first meta-analysis of systemic pesticides. It's a 4-year study worldwide, and we reviewed more than 800 peer-reviewed publications. This work was done by 29 independent scientists, This is important. They are authors, and this was done on a voluntary basis. And these scientists came from 15 countries representing 4 continents. And it was published this year in Environmental Science and Pollution Research. So this is a summary of this work, but I will go into detail a little bit on each subject. So I told you that the beginning of the story was the global collapse of entomofauna, and we decided to explore all the causes that were possible to explain this. So global warming, the presence of electromagnetic waves everywhere. But in fact, to explain such a phenomenon, you need to have one cause beginning at the beginning of the phenomenon, and one cause which is widespread over the globe. And one cause remains, one hypothesis remains, systemic pesticides. So it was at this stage still an hypothesis and not a demonstration. So we decided to investigate. In the second paper, we present, in fact, these systemic insecticides, the trends, uses, mode of action, and metabolites. I don't want to speak too much about metabolites. It's pretty complicated. But we studied the metabolites in animals, plants, water, and so on. I just want to show you 2 slides about this work. First, the global market of nicotinoids. This data comes from Japan. And you can see that after 1994, the launch of these products, the global market in Japan increased sharply. The scale is in tons. This is the time. And you can see also that there was one product at the beginning, then 2, 3, and more and more and more and more, all belonging to the same family, all being very similar to the others. And if you compare the global market in Japan here, in California here, just California, in Sweden, no, in Britain and in Sweden, it works very well to control pests. but it is used as it's a prophylactic use. So it's used as preventive and massive uses. So there is a lot of amount of this insecticide that is used. It's 40% of the global world market. These 6 molecules among the thousands of pesticides. Something very interesting also. Here, Bayer, that's a license from Bayer. They say, I prefer to read exactly. They say that when you have 2 insecticides of the same family, they act synergistically. So the effects are more than the sum of the individual effects, okay? Or, you remember, in pollen and nectar, you have several insecticides together at the same time. And I can show you something very interesting. When studying this insecticide in the registration document, you can find some information about the parent compounds, but usually there is no information about the metabolites. And you have to know that all these metabolites are as much toxic or more toxic, even more toxic than the parent compound. And you have this example here of cametoxam, all metabolites here, and one metabolite here is clotanidine, another product. You have seen the metabolite in the analysis of bees, for instance, dead bees. And this clotanidine has its own metabolism here. So, in fact, there is a long series of metabolites everywhere in the world. I can show you another paper we produced. It's about the environmental fate and exposure, of course, but it deals with the environmental fate in air, in soil, in water, in plants, because all these products have a long duration in environment, more than months, sometimes more than years in soil. So I just want to show a little section about pollen and nectar, because we are speaking about bees, and we reviewed all the literature about that, and we found that pollen has a mean contamination from 1 to 30 to 39 nanograms per gram. Remember that bees are sensitive to 0.1 nanogram per gram. And for nectar, now, we found a mean contamination from 1 to 73 nanogram per gram. Another paper on the effect of nicotinoids and phycolane on non-target invertebrates. So here's the list of invertebrates: honeybees, butterfly, diptera, hymenoptera, hemiptera, neuroptera, coleoptera, arachnida, houseworm also. I will show you just 2 things about terrestrial invertebrates and aquatic invertebrates. So, because I can't show everything, I decided to summarize. Here is a legend about exposure. Well, when there is no exposure, the score is 0 here. When there is a maximum exposure, the score is 4. And this invertebrate can be exposed by plants, by air, by soil, or by water. And about the ecotoxicological effects, they can be potential, or there is some evidence at high doses, or there is some evidence at moderate doses, or there is really evidence at very low doses. So, briefly, about pollinators, you will understand easily that they are exposed by plants, pollen, nectar, but also by the contamination of air, and to a lesser extent by the contamination of water. And the ecological effects are very important. They are maximum, not only for individuals, but also for populations. And we don't know about communities. Population, when I say population, is to say beehives in a large area. When I say communities, it's all the bees. And this is very important for food web support, of course. About terrestrial invertebrates, we found it that the main exposure occur by plants and soil. And about ecotoxicological effects, there are maximum about individuals and population, and we don't know about communities. It's also very important for food web support, for soil quality, soil formation. Aquatic invertebrates, there is a big impact on aquatic invertebrates. Of course, they are exposed by water. The effects were shown on individual, population, and also communities, means species of this, and some species are disappearing. The effect is not maximum, but occurs at those moderate doses. And this is very important for the food web support. Food web support, and not food we support. Sorry. So, now let's go to vertebrates. About birds. Birds are eating insects to raise their chick. Birds are eating treated seeds. So there is an impact on birds that are exposed by plant and by soil. Seeds here, plants, because they can use also pollen and nectar. We have found some effects on individual, probably some effects about population at the time of this publication, and this is very important for seed dispersal, pest and disease regulation, pollination, and so on, and for food also. And just after the publication of our work, there is a new publication here in Nature, and which realized the decline of insectivorous birds with high nicotinoid concentrations. This was observed in Netherlands, but this is probably the case everywhere in Europe. Now about fishes. They are exposed by water, and the effects are shown at the individual level and population level, and we don't know about communities. And this is really important for food also. Finally, we wanted to see what are the risks, the large-scale risk of the use of systemic insecticides for ecosystem functioning and services. And I just read this sentence. We have a broader evidence of the effect of ecosystem function regulating soil and water quality, pest control, pollination, ecosystem resilience, and community diversity. So this is not just a problem of species, even a problem of population. This is a huge problem of ecosystem services that is involved here. The last publication, because it's probably time to stop, the last publication was created because it's not sufficient to say you don't have to use too much pesticides, you don't have to use this systemic pesticide. You have to propose other solutions. And in fact, we wanted to test alternatives because farmers generally say, but what to do? We need pesticides to protect the crops. So we tested some alternatives to nicotinoids in two cases. In the cases of maize in Italy and in the cases of forestry in Canada. And I can show you this chart. In fact, in some cases, you need pesticides, and pesticides need to be used. But in most of the cases, I don't want to go into detail here, but in most of the cases, 90% of these cases, you don't need any treatment because there is no pest. The preventive treatment is not useful. And in 10% of cases, you can choose between biological control or chemical control. And in fact, this is a very recent publication from the EPA. It's October the 16th. In fact, EPA is examining the usefulness of these insecticides, which represent 40% of the global market. And they find it that they are not useful in this case for soybean production. So companies are selling very toxic pesticides for nothing in most of the cases. So now we go to the conclusion. There is a preventive and massive use of these insecticides because they are very toxic to invertebrates, and because they are also very toxic to vertebrates, and because there is a very high persistence in soil, in water, in air, in plants. All these properties are leading to a general collapse of biodiversity observed on invertebrates, threats on ecosystem stability, threats on food production and food security. That's why our task force, a group of independent scientists, we think that the present use of systemic insecticides is not sustainable at all in industrial country with intensive agriculture, of course. So we advise to reduce or suspend and increase the use of these pesticides and increase integrity pest management. Well, I would like to finish by many thanks to all my colleagues. I am not alone. I am alone here today, but I am not alone to work. My laboratory and my institution, and the foundation, which gives some support and financial support for all this work, is welcome if you want to participate. And I thank you for your attention.
So, do we have any questions from the audience for Dr. Bonmatin? If I may. Okay, please, Elizabeth.
More than honey.
It is not correct, really. We have contact with beekeepers in Australia, and they say bees are dying. There is massive dying of bees. So it's not correct. What is correct is that there is some conflict of interest in Australia. The guy who is selling the product for companies is involved in the assessment. So about the data from Australia, everything is wrong. But if you have a direct contact with beekeepers organization in Australia that will say to you that 1/4 to 1/3 of the bees are dying every year, and they have to renew the beehives in the population. Yes, it is correct because all these causes, pesticides, parasitism, and pathogens are acting together, but in some cases they are acting alone. Okay. About bees coming from South America, there is little differences, and they are more used to defend about parasitism. Okay, so in some cases there is less mortality in South America, but in other cases where the pesticides are responsible, the mortality is the same.
Also 30 to 40%. Those are of the colonies that are formed that year. What about from year to year? Do we start every year with the hope of a large number of colonies and then some collapse, or are we seeing a progressive?
No, the number of colonies worldwide is almost the same because beekeepers are dividing colonies, they renew colonies, okay? So we have at this moment a similar number of colonies, but the duration of this, there is a real threat on honeybees and on the number of colonies because beekeepers are just they abandon their work. Okay, there are less and less beekeepers in Europe.
What percentage of all bees live in human-formed colonies for agriculture versus number of bees in the wild? Are humans now, in some sense, the dominant beekeepers, or are there more bees in the wild?
Well, in fact, I could give another answer. We study and we use 4 species about honeybees and bumblebees, and there is 25,000 species. So what is true for 4 species is probably true for the other other species. And I would say that when bees of beekeepers are disappearing, probably all the wild bees, solitary bees, are also disappearing. And nobody renew these wild bees.
I have a question. The DuPont Corporation that makes a lot of commercial seeds, they also make a Roundup chemical called Roundup that is, and a lot of their wheat and things like that is resistant to Roundup. And as a result of which we have seen exponential increase in grain production that is resistant to weeds. And instead of you can't beat them, join them. Is there any way genetically modified you can make the genes of bees resistant to these toxic chemicals, these nicotine insecticides? Because after all, bee is an insect.
Yes. In my talk, I was focusing on insecticides because they have the most insecticides kill insects, and bees are insects. Okay, so the most dangerous pesticides for bees are insecticides, but also there is herbicides, fungicides, who can act together with pesticides, with insecticides to kill bees. About your question, some researchers are trying to find some species of bees which would be resistant to pesticide. But this doesn't solve the problem of wild pollinators at all. This could solve the problem of beekeepers producing honey, but not the problem of wild pollinators. So the problem of pollination.
Yeah, thanks for the talk. It goes a little bit in the same direction of the previous question. So we talked about insecticides, but I also heard that genetically modified plants could affect pollinators or the mortality of pollinators. How many studies are there about, or are there any long-term effects of genetically modified organisms on those pollinators?
There is few studies about that. In fact, there is no clear demonstration that the toxin produced by GMOs can impact honeybees. There is some clues, but not clear evidences. Actually, it's impossible to produce a pesticide, an insecticide, which kill pests and not non-target insects. It's impossible. They act on the central nervous system. The central nervous system is common to all insects.
But as a short-term measure, are there any technological alternatives to pollination that they serve? You know, the 50 billion euros worth of service that they do. Are there any mechanical or other ways of?
I know Chinese people are doing that by hand, on trees, for instance, but it's not a serious solution. Thank you.